my computer's a little bit unstable, so I hope that this is going to work. I am a chemist of sorts. Wish I was an artist. And I'm going to talk about some things that inspired me to do science, I suppose, in general. This particular field of research all began really with Alan Turing, probably, in the 1950s, who you probably know better for trying to, well, for solving the Enigma code during World War II, and he's the father of computation in some respects. But one of the problems that he was interested in was how do you get the growth of form, so the growth of structure in nature, and it's called morphogenesis. And basically he took some chemical reactions in a computer simulation, and he distributed them spatially and connected them together. And what you can see on the left-hand side there is some equations that he solved on the computer. And what you see is for certain sets of conditions you get the growth of patterns. So this is computer simulations. The one on the left is not working. Oh yeah, okay, there we go. So for different sets of conditions you can get the appearance of stripes and spots, and this is basically just a chemical reaction distributed in space. And so people were of course fascinated by this idea. I wanted to see if they could reproduce this in the lab, doing chemical experiments, and it took them a very long time actually. And Alan Turing's original motivation was mainly, I think, the structure of Hydra, which is a kind of a sea creature, and it's got tentacles. But people, of course, immediately thought about animal markings and stripes on zebra coats. And the one on the left-hand side is an animal marking, and the one on the right-hand side is a chemical reaction in a lab. And I should say that probably this is about 100 microns, so this would be about 1/10 of a millimetre, so like tiny, a tiny domain. This is blown up a little bit. So let's reveal what animal this is. This is the one on the left is a fish. And the one on the right is this chemical reaction performed in the lab in a special reactor. And then you can do things like you can take these chemical reactions and you can do them on surfaces. And these are some simulations by Thomas, who's working with me at the moment in the chemistry department. And so you can take the reaction and you can do it on a tube, and then you can have these kind of stripy patterns and spotted patterns on the surface of the tube. And the size of the domain influences the patterns that form. So if you've got a really tiny domain, you will end up with stripes, as we can see here. Whereas when you grow the size of the domain, you might end up with spots, or you change your parameters and you end up with these more complex looking patterns. And so some animals have spotted backs and stripy tails. And often there's certain types of snakes as well, whereas it tapers off, it goes from being a spotted back into a stripy tail on the tail. So that's basically, this is just a chemical reaction, and then it's obviously more complicated in the real biological system. And we're trying to produce these things in the lab at the moment. So around a similar time in the 1960s, there were some guys working, one guy, Zavotinsky, working on cycles in biological systems, and he was trying to create a lab-based cycle, and he mixed a bunch of chemicals together, and we can see what happened in this little clip here. So basically he took these chemicals, they're fairly simple chemicals, then nothing special about them at all. You mix them all together, you add a catalyst which makes it bright red. This is an iron-based catalyst, the same stuff that makes your blood red. And at some point you get a change in colour. It's great that this is a video because I know this is going to work. There you go. This is actually this is a YouTube clip, and this is a bunch of Japanese scientists, and I particularly like this clip because they get really excited about it. And so the weird thing was that it turned blue, but then it went back to red again, and then sometime later it goes blue again, and then it goes back to red again. And it doesn't do that forever, and I think chemists were really shocked by this discovery, because usually when you mix chemicals together, it changes colour and that's it, that's the end of the reaction. And when this poor old chap here, this Russian scientist, tried to publish his work in the 1960s, the chemistry community said this is impossible. You know, chemical reactions finish, they don't turn back to the original thing and carry on. So I've actually brought these chemicals with me, and later on in the break we can mix them up and I'll prove to you it is real. It's not a figment of my imagination. So the work was taken up by another Russian scientist, Belus. So this is Belousov, and Zhabotinsky took up the work, and then eventually it was accepted by the chemical community sort of in the 1970s. And the reason why people then got very excited about it is because you can do it in a Petri dish. Doesn't sound instantly exciting, but then you get patterns like this. And so this is a Petri dish, this is about that size. And these are propagating structures, and I can possibly show you in a movie if my computer's not angry. Yeah, it is. So here you can clearly see the formation of it. Maybe not. We'll come back to that. So yeah, you basically take those chemicals that were changing colour and you put them in a Petri dish, and these patterns just appear spontaneously. And I'll show you it later in an actual experiment, and then you'll see. And it turns out that you can see spirals on all scales and in all different kinds of systems. So this is that BZ reaction in a Petri dish. This is cells. This is a slime mold system that sends out chemical signals that turn out to be spiral patterns, a similar sort of length scale, pretty small. That's a bit of nerve tissue. So things in nerve tissue propagate, chemical signals propagate outwards as spirals. That's combustion on a crystal. Spirals. We've even got, this is actually combustion, this is a flame, a spiral flame, this is a spiral galaxy. I'm not saying they're all due to exactly the same processes, but they're certainly prevalent. And you can also do other crazy experiments with the BZ reaction, which is what a lot of people then spent very many years doing. So you can do the reaction on gels, and you can influence the pattern formation using light, and then you get these kind of interesting time oscillating structures. You can also get, again, this is probably isn't going to work. Let's have a look. So this is a slightly different, oh yeah, okay, you can see this. This is a slightly different chemical reaction, but you can, under certain conditions, what you see is these are domains where a chemical reaction has happened, and the way it spreads out is it forms a spot, and then the spot splits, and then each of those spots splits, and you get this pattern forming outwards with each spot splitting in time. This is speeded up a little bit. So that's another chemical reaction that can show interesting sort of patterns. And then, well, if you take heart waves, fireflies, another example. This is an example of oscillating processes that occur in cellular systems, and we were sort of interested in the idea that you can then take cells that have these oscillating chemical reactions and connect them together and get new structures and patterns, and sometimes they're really important, like in your heart, you want your heart to beat as one unit, and that's all these connected cells that send signals. Fireflies can actually synchronise their activity. You can just about see on the right hand side there, this is a tree full of fireflies. There's only one species of firefly that can do this, that can actually see its friends, and it ends up synchronising its flashing with its friends. I think there's something like maybe 25 species, or maybe more than that, and there's only one that actually synchronises. This is that slime mould again, so this is spiral waves in a biological system. And these spiral waves are quite useful to this organism because it uses them when it's starving to signal it's time to form a slug and get out of here. So it basically crawls, all of the individual cells crawl together, form a mound, and then form a slug, and then the slug crawls off, and when there's lots of food, it plants itself down, makes new cells, and the whole life cycle of this little mini organism starts over again. So spiral waves are useful to some organisms. And this is actually that BZ reaction again. This is on some catalytic particles. So this is, we took the reaction and tried to make it local, to make cells out of it. And then you can get all kinds of fun patterns. Again, these are really small, so that's like a millimetre, again, this whole thing. And then you can get waves and you can get spirals. And then you can get quite complex looking structures. So that's kind of some things that we're playing with. And then we're trying to take these reactions and do them in things that you can actually eat. This reaction doesn't oscillate, but this is a reaction that occurs in your body. This is an enzyme catalyzed reaction. Urea, which is a chemical that's really common in your body, and you can it's produced in your stomach, and then there's a little bacteria that actually uses that urea to raise the pH, to make the environment more friendly for it to live in your stomach, and it's the one that eats your stomach wall and makes holes in your stomach wall. However, this reaction is really simple, and these are just little particles of basically that reaction occurring locally, and yes, they are edible. Although I'm not suggesting that anybody ate these. So yes, that's some waves and patterns from our lab, and that kind of concludes my talk. And I should say that possibly some of these things are useful to some people, but I just still like the patterns.
There are a few minutes for questions if anyone would like to ask questions.
Your mathematical models are mostly based on differential equations. Have you explored integral differential equations for modeling these phenomena at all?
Well, these equations are integrated as a function of time, and that's how we solve them.
But they don't have an integration step in the time step. You integrate them to find solutions.
Yes.
Right.
Yeah.
Have you looked at all at the Boltzmann type equations?
The Boltzmann type equations.
Used to model gas kinetics, but they've been
Yeah.
adapted to model biological phenomena and chemical reactions in non-homogeneous media.
Oh yes, okay, yeah. No, we don't use those types of equations, but yes, some people do. Yeah, because you can model these reactions also like cellular automaton, where a similar sort of idea, and yeah, you can model them on a molecular level with an event probability. You can do it in that way, and some people do and have in the past, but yeah, we don't. We have a continuous domain, and we solve them in that way.
I have a question, and I know that there's about a minute left. Do you understand anything from the fact that these patterns seem to replicate on different scales? I mean, when you see that, what do you think?
Yeah, I think it's really interesting. I don't know. I'm not sure about the mechanisms of spiral galaxy formation. Like, I don't know whether you can think of it in the same way. I'm not sure. But certainly, just the appearance of a spiral occurs in so many different places in nature. I mean, we were even thinking of one the other day where trees, when they die sometimes in the middle, and you have a hole, the tree curls in and forms, it grows as a spiral wave inwards. So many phenomena are just naturally spiral waves on all different scales. And I think but nature has this tendency to repeat patterns, doesn't it? And they're not always from the same mechanism, scientifically. Yeah.
So, Annette, what's the parameters that allow you to tune these patterns across length scales? Is that too simple a question in terms of the answer's not that easy?
No. When one of the massive things is how you connect these things together. So you have a reaction, or you have something making something, and then it has to spread outwards, and it's the time scale of that process, it's the and it's the transport part of it that governs the length scale. So usually you're talking about diffusion, and that has a certain characteristic time scale associated with it.
So slower diffusions, bigger patterns, or?
Smaller.
Smaller patterns, okay.
Bigger diffusion, faster diffusion, bigger patterns.
Okay.
In the context of spiral galaxies, I don't really know what, but there's some kind of transport mechanism involved in that, and it's probably, related to that, yeah, indeed.